Good afternoon, everyone. I would like to thank the team of Dr. Lau and Dr. Punsalan for inviting me to be part of this series of webinars that discuss the chemistry behind different phenomena that are happening in our oceans. I would also like to thank everyone in the audience for participating in this talk despite the seductive Christmassy nature of this afternoon. So my talk will be the connection between ocean acidification and the organism that I've been studying for the past 8 years, the mangrove crab. To be honest, when I was asked to do a talk about chemistry in the ocean, I really didn't know what to talk about. You see, I'm neither a marine biologist nor a chemist. My primary training is in molecular biology and upland ecology, and my research has mostly focused on species identification, population genetics, and upland rehabilitation. But for the past 8 years, I've been involved in several projects about the mangrove crab, locally known as the alimango, which brought me closer to the shore and also had a lot of chemistry in it. But why study mangrove crabs? Why are we so interested in the alimango? Well, this 5.2 billion peso industry serves as the primary source of income of more than 15,000 families in the Philippines. And we are only second to China in terms of production. The initial goal of our research was to identify the population genetic structure of the alimango in the Philippines across the 7 known sites with wild populations. But what is population genetic structure? The genetic structure of a population is characterized by the number of subpopulations within it, the frequencies of the different genetic variations or alleles found within these organisms, and the degree of genetic isolation within each location where the subpopulations can be found. Studying population genetic structure can actually help us predict how past characteristics of interest, such as, let's say, vulnerability to disease or heat tolerance, could arise and spread in natural populations. Using different morphological and molecular markers, we were eventually able to detect an east-west population structure in the Philippines, with the Kuroshio current connecting the populations in the Pacific Ocean and the downward movement of the West Philippine Sea connecting the populations on the west. Execution of this study brought us to different places in the Philippines, and as we traveled across different fishing communities, we became aware of other problems. So as a complement to this study, we were also able to create the application called Crabifier to identify juvenile crabs to species, to help our fishers verify crablets sold by traders, and to help them identify their preferred species. Crabifier, or any mechanism to detect species of juvenile crablets, are important because in the Philippines, out of the 3 species of mangrove crabs, only Sila serrata is preferred due to its fast growth rate and larger sizes upon maturity. Inconveniently enough, their habitat range overlaps all over the world, making any captured batch a random mix of all species. And our hatcheries have yet to produce sufficient crablets of a single species to fulfill local demands. Now, the limitations in the usefulness of Crabifier to our fishermen is not its accuracy, but the fact that you need a cell phone camera to use it. So, we are continuously on the prowl to find non-tech dependent means to identify juveniles to species. In order to facilitate that, we went to the source themselves to look for traditional methods of juvenile species identification. And what keeps popping up is the color of the ventral carapace of the crab. Our fishers insist that a reddening in the joints of the sternum of the crab indicates that it is Sila olivacea, the smallest species that is locally called kulahan. We tested this technique against molecular markers, but it only showed that the reddening of the sternum happens in all species of the crabs. We then initially thought that the observed reddening might actually be caused by warming waters. After all, we do observe a color change reaction in crab shells after we cook them. But this is actually due to the astaxanthin pigment being released by the crustacyanin molecules holding them. Crustacyanin is a special molecule that masks the red color of astaxanthin in live crustaceans. But with heat, it is destroyed, and thus, it releases astaxanthin, and the true color of the crab, lobster, or shrimp carapace is revealed. But going back to our initial problem, the sternum of the crab has little to no crustacyanin, or astaxanthin. And so, we are led to think that something else might actually be interfering with the carapace formation of our crabs, which finally led us to be interested in ocean acidification. Ocean acidification is the less popular sibling of warming ocean waters in terms of the consequences of man-made global warming. You see, with our overproduction of carbon dioxide, there is a consequent overabsorption of that molecule by ocean water. Chemical reactions occur, as shown on screen, that reduces the seawater pH to acidic levels. So, how is this connected to our crabs? Now, calcium carbonate minerals are the building blocks for the exoskeletons and shells of many marine organisms. And the ocean is rich with it due to constant processes of erosion and decomposition. At normal pH, carbonate ions in the presence of calcium turns into calcium carbonate that can be used up by marine organisms, including our crabs, for their shell development. But because of elevated levels of carbon dioxide, the pH of our oceans has become too low, and the presence of excess carbonic acid has captured free carbonates, preventing them from being used by our marine organisms. Now, this doesn't seem to be the answer to our red-bellied crabs, so we're looking at diseases for this now. But some things are matching up. Our fishers are actually complaining of weak crablets, pale and thin-shelled from Cagayan, the northern Pacific, where we used to record the most robust populations in terms of heat and salinity. You see here the ocean acidification map of the European Space Agency that records the area of Cagayan and the northern Philippines to be experiencing the most levels of ocean acidification. And so, our team seeks to monitor ocean water pH in select areas with wild populations, and to create simulations to determine at what pH and when during the crab's developmental stage it is most affected by the fluctuations seen in our waters. The consequences of climate change are fast becoming real, and though these feel like accidents or catastrophes waiting to happen, we are hoping to find vulnerable spots that will need protection, and we are hoping to create interventions to avert disaster. So thank you so much for listening to my short talk, and I hope it was able to open doors of interest for you to be interested in ocean acidification or the study of our mangrove crabs. I am now open to your questions, comments, and suggestions. Once more, thank you very much.